This is the last section of chapter 13, and it is on genetic engineering. So, genetic engineering is the technology that involves introducing exogenous DNA into an organism's genome. What exogenous DNA is it's manipulated, removed from another organism, or artificially created DNA. And so, if we are altering the genome of an organism in some way, manipulating it with our technology, that is genetic engineering. In the example that I have shown here, we have the GFP protein, the green fluorescent protein, and it has been introduced into mosquito larvae. And so you could see these larvae are glowing under the stereoscope. GFP is an interesting protein. It is synthetic, and we've introduced it into a variety of organisms. You could actually buy glowing fish at the store for about $10, and so you could buy your own genetically modified organisms and have them in your fish tank. They've also introduced this protein into cats, just to show that they could. They had glowing mice. You can't buy glowing mice or cats, though. A lot of students ask at this point, well, can you make a human glow? Hypothetically, yes. But a long time ago, it was determined that would be considered unethical, and that we're not to modify the human genome in any way unless it's to cure a disease. And so there's a variety of tools that we implement in genetic engineering, and the first type is called restriction enzymes. And so there's a variety of different types of restriction enzymes. One of the most famous ones is EcoRI. It's a specific type of restriction enzyme. And what restriction enzymes do is they cut DNA sequences at specific locations. And so EcoRI cuts DNA at GAATDC. And so when it does that, it creates what we call sticky ends. And so if we take our DNA, it's cut by the restriction enzyme. Remember, enzymes are proteins. It's going to create these two sticky ends for which it can be used to fit another DNA sequence, kind of like a puzzle piece would. And so this restriction enzyme results in sticky ends, but you could also get blunt ends. Blunt ends are a little bit easier to combine with other forms of DNA. And so by cutting the DNA, it's the goal that we could introduce new forms of DNA into that cut. And so that's what restriction enzymes do. And so we could use restriction enzymes for recombinant DNA technology. And so if we take a vector, and so vectors are typically going to be plasmids, but they could also be viral DNA if we were going to use a virus, or sometimes even the plasmid of yeast. But here we just have plasmid DNA from a bacterium. And so the first step is we're going to use a restriction enzyme. And the restriction enzyme cleaves the plasmid, the DNA, at a specific location. And then that allows us to insert our own DNA, which we've taken from another organism, or we've artificially created or manipulated in some way, and we introduce that. So we put the restriction enzyme in there, cuts the DNA, we have our exogenous DNA introduced, and then we combine it with DNA ligase. If you remember DNA ligase is the enzyme that comes along and it links all the Okazaki fragments so they're continuous. And so we use that same enzyme, we put it in with the exogenous DNA, the cut plasmid, and what the DNA ligase does is it combines it, it reforms the plasmid now with that new strain of DNA, that new exogenous DNA that was introduced. And so at that point, we're ready to clone the DNA. And so to clone the DNA, we have our plasmid, and also part of the recombinant plasmid, so part of the recombined, so that's kind of what recombinant means. It means recombined. Part of that plasmid is going to be, it's going to make a protein that is resistant to the antibiotic ampicillin. Ampicillin kills bacteria because it is an antibiotic, so antibiotic, anti-life, antibacterial. And so if we introduce the ampicillin in the plasmid, that's going to play a specific role in the gene cloning as well. So I'll get back to that. And so we have these synthetic plasmids, these artificially engineered plasmids that we've created through the ligasing and the exogenous DNA. And then we have bacteria, and we have the bacteria transform those plasmids. And so the process of transformation is when the bacterium is going to uptake that DNA. It's going to actually take the DNA in itself. And that's kind of why bacteria have plasmids to begin with, is so that they could take plasmids from other bacteria, and it's kind of like their form of exchanging of genes. And so most organisms undergo sexual reproduction, and that's how they're going to exchange the genes and get more genetic diversity. Bacteria, they are going to undergo transformation in bringing the plasmids of other bacteria. And so the first step, we make the plasmid. The second step, we have the bacteria transform, and then those bacteria will replicate, and they'll replicate, and they'll replicate. Some of the bacteria, they will replicate and they won't have our plasmid in them, and some of them they will. And so if we zoom in on this picture, you can see some of the individuals have the exogenous plasmid, the exogenous DNA. Some of them don't. And so how do we separate these bacteria from the ones that have it and the ones that don't? And so what we do is we introduce the antibiotic ampicillin. And so if we introduce the antibiotic ampicillin, it'll kill all of the bacteria that do not have our exogenous DNA. Because remember, we put in that ampicillin resistant piece of the plasmid in there so that the bacteria that did transform, they did take that plasmid within themselves, or they were duplicated from the individual that took it in themselves, they will be resistant to the ampicillin. So that's a way we could separate out the ones that have transformed our plasmid and ones that didn't. And so that is called gene cloning. That's how we could take exogenous DNA, scale it up, and get a lot of copies of it, is through this process. Once we have a lot of our recombinant DNA, this technology is known as biotechnology, and it's used to find solutions to problems through making transgenic animals, plants, bacteria, and we use them for research in medicine and agriculture. And so we can have transgenic plants, we can have transgenic animals, transgenic bacteria, transgenic fungus, so on and so forth. And by having these transgenic organisms, they're called transgenic because they have the genes from another organism or artificial genes within them. We could study a lot of different things. So, for example, we can make transgenic plants that are resistant to pests or disease. And so you could see this scientist is looking at a transgenic plant. The one on our left shows the leaf has not been eaten by probably some type of caterpillar or other type of pest, and the one on the right has. And so this one on the left is a transgenic plant. It has been modified so that it is resistant to that pest. Other plants that have been modified, we could use them to reduce allergic reactions in humans. We could have them increase vitamin and mineral content in our crops. We could have our crops be resistant to extreme weather. We could have them help produce vaccines or biodegradable plastics. For transgenic bacteria, we can design bacteria to produce insulin for diabetic patients. We could have them produce growth hormones. We could have them help slow the formation of ice on crops for the bacteria. We could even have, we could design bacteria to help us clean up oil spills. We had that occur in the Gulf of Mexico. We introduced a lot of different types of transgenic bacteria and to help us try to clean up the environment. And then we could also design bacteria to help decompose garbage at a faster rate so that we could be using them in landfills. There's a lot of different types of biotechnologies when it comes to transgenic organisms, just taking genes from one organism and putting them in another organism. But cutting edge technology is actually artificial gene synthesis or DNA printing. And so what we can do is we can actually print DNA sequences. And so a scientist might want to be studying some type of gene, and they want to see if they could alter that gene in some way. So they'll actually go on to a website and they'll order a specific sequence from a lab, and that lab will print them that sequence of the DNA, and they could use that to see what type of protein alterations they can make, or different types of changes in expression for whatever transgenic organism that they're studying. And so this has a lot of advantages. It's cheaper than traditional recombinant DNA technologies. Instead of having to find the gene in another type of organism, we could actually take the original gene and alter it to see what we could do with it. We could obviously create custom sequences, and furthermore, we can incorporate unnatural base pairs. And so naturally, we have 4 nitrogenous bases that exist in DNA, but through this technology of DNA printing, we can incorporate additional letters. There are DNA sequences that have been artificially designed that contain as many as 8 different letters, and so that just opens the door to many possibilities. The issue with artificial gene synthesis is that it's extremely error prone, because the way that they have to do it, they print one side of the DNA, and then they print it from the 3' to the 5' direction, and then they have it scaled up through the PCR and using DNA polymerase. But the problem with it is, because the way they have to print it, it's very error prone. So it's very difficult to get any DNA of any substantial length without having lots of errors. And so right now, where the technology is at, it has pretty much a 200 base pair maximum. So that's like the maximum length. And since one gene is typically around 100 base pairs, we could pretty much get one gene at a time printed. Another issue is that custom DNA can be very dangerous, in that if someone knows what they're doing and they wanted to create something harmful, like a harmful pathogen, like even like a virus, they could technically do that. They could print their own virus, their own custom virus, and that could be used as a bioweapon. And so these labs that are printing these DNA sequences, they actually they look over the sequences to see if they're associated with any harmful viruses or any other pathogens before they're printed. And this technology is advancing very quickly. It's the hope that eventually artificial intelligence can help us analyze these sequences before they're printed, so that no malicious sequences are printed. One of the most powerful DNA tools that we have in our toolbox is the CRISPR-Cas9 system. So scientists were studying bacteria, and what they realized about the bacterial genome is that pieces of their DNA actually matched this DNA of known viruses. And so they said, okay, well, why do these bacteria have viral DNA as part of their DNA? And so eventually they figured out, well, bacteria do not have the same pathogen fighting systems that eukaryotes have. So you and I were multicellular organisms, we're eukaryotes. When there's a pathogen that our body's exposed to, when we overcome the pathogen, the virus or the bacteria, we develop antibodies that help us recognize that if it comes to our system again, so that our white blood cells can take care of the problem. Prokaryotes can't do this because they are single-celled. They don't have white blood cells. They don't have antibodies. And so the mechanism in which bacteria address this is through their CRISPR system. And so if a virion injects its DNA, remember, there are very complex virions, bacteriophages, that attack bacteria specifically. And so if we have that viral DNA, it gets in the bacteria. If that virus, if that type of virus has infected that population or that species of bacteria at some other point, that bacteria is going to have part of its DNA stored in its DNA. And so the bacteria has these genes called, we just call them Cas genes, CRISPR associated genes, and they're going to make these Cas proteins. And what these Cas proteins do is they use the CRISPR region of the bacterium's genome, and it uses it to identify the viral DNA. And so if this viral DNA has already been inside this type of bacteria before, the Cas protein is going to recognize it, and it's going to destroy it. It's going to go and just cut it in half, therefore deactivating it. And so that's the first type of scenario for CRISPR. The second scenario is if the virus attacks it, and then we have the viral DNA, and that viral DNA is not familiar to the bacterium. Well, the bacterium might still be able to take out that viral DNA and might be able to fight it off. If it does, not always, but if it is able to do that, it's going to then take that viral DNA and incorporate it in its CRISPR system, so that all the progeny, all the offspring of that bacteria, once it divides for many generations, every bacterium that's developed from that bacterium thereafter will have it in their CRISPR system. And so what we find is all of these bacteria have these mini databases of all these viruses, and so that they can be recognized, and these Cas proteins can be transcripted or translated, can be translated, and then they could just destroy the virus. And so we discovered how this CRISPR system works, and we said, okay, well, why don't we modify that CRISPR system, and why don't we make it so that we could put in our custom RNA for that Cas protein, and then we could make the CRISPR protein cut whatever DNA we want at whatever location we want, and then we can incorporate whatever DNA we want. And so we've modified the CRISPR-Cas system, and we use this Cas9 protein. In this Cas9 protein, we are able to modify the RNA that the Cas protein is using. Instead of using part of the CRISPR system, it's just using the custom RNA that we're giving it, and it's identifying that part of the DNA sequence. It's cutting the DNA sequence right there, and then whatever custom DNA we have to implement into that system, it's going to be put right into the DNA. And so we are using this CRISPR Cas9 system to specifically locate parts of DNA sequences in whatever organism we want, cut it on site, and then put our new DNA, our new custom DNA, in that location. You might think that this sounds similar to restriction enzymes, and it is. It's very similar to restriction enzymes. But the difference here is that for restriction enzymes, we have to engineer the enzymes to cut the DNA at specific locations. This is a very complex and very expensive process because, as you know, proteins are very complex molecules. They have 4 levels of structure. For the CRISPR-Cas9 system, the Cas9 protein already exists. We're just swapping out the RNA that it's using to identify the DNA sequence that it's going to cut. Therefore, the hard part, making that protein, it's already done. We're just isolating the protein from bacteria, and we're giving it whatever RNA sequence we want it to identify, and then it's going to do all of the work for us. And so the difference between using restriction enzymes and CRISPR-Cas9 is this. In CRISPR-Cas9, we're engineering RNA, and it's a very quick, easy process because RNA is a very simple molecule compared to proteins. When we're using restriction enzymes, we're engineering entire proteins. It's very difficult to do that, and it's very complex to do that. So the CRISPR-Cas9 system allows us to, it's pretty much the ultimate tool to cut DNA at specific locations and implement whatever DNA at that location we would like. And so the future for CRISPR-Cas9 is gene therapy and gene editing. Gene therapy is a technique aimed at correcting mutated genes that cause human diseases. So if we insert a normal gene into a chromosome to replace a dysfunctional gene, it's our hope that that cell will then be repaired. And so if we do that in all the cells, then we could fix different types of mutations that are known, which, remember, we are storing as in databases when we're studying bioinformatics. And so CRISPR-Cas9 has a lot of potential. You hear a lot about it in the decades to come. It is the most cutting edge biology that we have. So hopefully you learned something.